First of our experts on nematodes today, Daniel Leduc from the Institute of Water and Atmospheric Research in New Zealand, perhaps one of the furthest travelled members of this workshop. He's going to be talking to us about the deep sea nematodes, focusing on abyssal and vent environments. Over to you.
Thank you. Good morning, everyone. It's nice to be back in Ghent. Today I'll be talking about nematodes, and you'll notice I didn't use the word Meiofauna on the slide, and that's because nematodes span Meiofauna and macrofauna size classes, especially if you use a 250 micron mesh. It's a big topic, and there's a lot to cover, and I've left things out, hopefully get the most interesting things in. I'll begin by talking about the taxonomy itself, move on to talk about diversity trends in the CCFZ and vents, and then talk about ways in which we could usefully use the nematode data to inform mining in the future. It's all very hypothetical, obviously, it's early days. So what is a nematode? Nematode are small worms. They're not segmented, and they molt, which some people don't know about. They molt between the different juvenile stages or larval stages. So in that way they're related to other animals that molt. The free living forms have separate sexes, you have males and females. And I put this diagram up there just to show you basically that there's a fair bit of morphology going on. A lot of people's experience of a nematode is a small thread under the microscope, and when you look at them closely, there's actually quite a bit going on, even though the basic body plan is a tube within a tube, in a tube between the being the intestine and the outer one being the cuticle. That's nematodes in a nutshell. They make a living in quite a wide variety of ways. Obviously, there are free living forms that live in marine sediments, but we also get them in soils, freshwater, and so-called extreme environments like seeps and Antarctica. There are parasites, obviously, sometimes are vertebrates, like fish, ourselves, invertebrates, like crustaceans, and plants. And some of them do a little bit of both, like this example up here, which belongs to the Benthimermithidae. And they are parasites of invertebrates in the deep sea as juveniles, and as adults, they emerge as adults. And sometimes they turn up in core samples, which is why some of you may have seen it before. Nematode classification is still changing, so there are various versions of this classification. But basically, we have 2 classes, 3 subclasses, and various number of about 26 orders. 12 of them have marine representatives, and 11 of these have representatives in the deep sea. Of these 11 orders, we have 8 which represent the bulk of all the nematodes you'll find in deep sea samples. There's another 3, one of which I've already mentioned, the Benthimermithids, which have known parasitic stages or suspected parasitic stages, and they are pretty rare, but they do turn up every now and then. An interesting feature of nematodes is that families and the majority of genera are distributed all around the globe, and also from shallow water to deep waters, which is nice because that means what we learn from the shallow water species we can apply to some degree to the deep sea species or deep sea taxa as well. Species, however, have a much more restricted distribution. They're often not widely distributed. Sometimes they are. There are a few cosmopolitan species in shallow water that have been confirmed, for example. But generally they have a more restricted distribution. And the thing is, identifying a nematode to genus level is no more tricky than identifying a nematode to family level. So as a result, a lot of the researchers out there take samples and ID nematodes to genus level. And one of the reasons it has been possible, especially in the last few decades, is because of the publication of monographs like these. If you've ever tried to identify a nematode, you'll know about these. These are 3 monographs published by the team at Plymouth. And what they do is provide pictorial keys to the genera. And the genus has a worldwide distribution, therefore people from all around the globe have been using it, even though these monographs focus on the British species. It's been really good. Myself, I found it really useful when I was doing my PhD, but it's starting to get out of date a little bit. They were published between 1988 and 1998. Although I'll still use it today if I'm confused about a nematode as a starting point. The other thing is that the deep sea genera are not so well represented. However, thankfully we have a newer resource which was published last year, the Handbook of Zoology and volume on nematodes. And this is quite a nice book, a big one. It covers all of the orders. And for each order, it includes all of the genera and all of the changes that have been made since the monographs, the Plymouth monographs have been published, have been put in that book. And for a lot of the orders, you get a key to the families, or the genera, or both. Sometimes it's a dichotomous key, sometimes it's a pictorial key. But all of the diagnoses for each genus that exists is in there. So that's really great. So what I do sometimes now is, if I need to identify something which I have no idea what it is, or maybe a beginner might do this, they will use the monographs, the Plymouth monographs, but then they should move on to this and figure out whether their genus identification is correct, in case names have changed or something like that. As for the primary literature, moving on to species identification, there's not that much out there for the CCFZ. So this is an exhaustive list of all the primary literature describing nematode species in the CCFZ, as well as the Peru Basin, which is about 5,000 kilometers away. These resources are on the Dropbox folder, so you can get them all there. Yes, so there's 2 people who have pretty much published all of what we know about nematodes in the CCFZ, Milutin and Meiofauna. There's a problem though. There's one resource which has the bulk of all the species known from the Distal Pacific, and that's the thesis by Busseau, which was published in '93. And technically this does not represent a published work in the eyes of the Code of Nomenclature. However, all the taxonomists, including myself, have been using it as if it was a published work. In fact, you can't publish a species description without being told by the reviewers that you've got to take this into account, so you can't escape it. So we need to fix this problem, the taxonomists, and one way to do it would be to republish it and make it a published work. But this has got about 100 species descriptions in it. And again, this is on the Dropbox. For bents, the list is very short as well. It's only 6 papers describing all of what we know about species diversities in bents worldwide. Again, it's on the Dropbox. Now, I did talk about taxonomy a bit, and I don't want to go into the details of morphology. I'll just give one example because it's a relevant and often useful example of morphological trait used for looking at ecology, and that's buccal cavity. So the buccal cavity can tell us about the taxonomy, but also what the animal does for a living. You can look at the size and shape of the buccal cavity, as well as the presence or absence of teeth, the size and arrangement. And then we can decide or have a guess as to what nematodes feed on. So, for example, this one up here has a very small buccal cavity and probably feeds on bacteria. So these are called bacterial feeders, or others call them selective deposit feeders. If the buccal cavity is a bit bigger, it's cup shaped or funnel shaped, they're called deposit feeders or non-selective deposit feeders. So they feed on detritus, whatever can fit in there really. And there's no teeth in these 2 examples. Buccal cavity can be larger. It can be, in this one, the top one, Elzalia, the buccal cavity is cylindrical. It's cuticularized, but there are no teeth still. And some people have I've made observations which suggest that they feed on ciliate or protozoans, protists. Again, it depends. Schemes differ somewhat between people. The one there is Acanthalamus, that's a deep sea genus. It's got a funnel-shaped buccal cavity with teeth, and that makes it a epigrowth feeder or epistrate feeder. So they're supposed to feed on algal cells, pierce the contents with the teeth, and then feed on the contents of say, a diatom or something like that. And then the buccal cavities get bigger, and the teeth get bigger as well. And you have things like facultative predators, which can feed on other nematodes, but also on other things like organic matter, detritus, etc. And the true predators like Gamenema, which have very large mandibles or teeth, which can move around and be used to chomp on other nematodes. And you can use all this morphological information to get an idea of the structure of the food web. You can look at the whole assemblage, say what is the proportion of microbial feeder, or what is the proportion of predators in that sample, and what does that tell me about the food web? And then you can compare different sites, and you can look at impacts before and after. The problem is, however, that all of this is based on shallow water species, and we're only guessing that the deep water species do similar things. We can look at gut contents to confirm or see if our system works. But that's a nice way, easy way to look into the ecology of nematode assemblages. Speaking of ecology, I'll move on to the diversity and ecology of nematodes in CCFZ and Ghent. I'll just quickly put that into a context by looking at the big picture. Now, you probably know already that nematodes are the most abundant animals. They're not animals that don't include forams. Their abundance, like any other organism, decreases as you go deeper. So as you get into the 4, 5, 6,000 meters, abundance is quite low. In the abyss, it's usually less than 1,000 individuals per 10 square centimeters. But it varies quite a bit. In the abyssal Pacific, the densities are a lot lower, at the lower end of that range. Like about a few tens to a couple of hundreds nematodes per 10 square centimeters. So that's good, makes it easier. For vents, densities are usually low as well. A couple of hundred individuals per 10 square centimeters, although densities can go up to about 1,000. But densities are normally low as well, which is probably makes life easier for us. We can do the same thing for diversity. And these graphs, by the way, are just data from all over, from the literature all around the world, including some from New Zealand. And they go down to the bottom of the trenches. Diversity also decreases as you go deeper. And diversity here is expressed as the number of species you'd expect in a hypothetical sample of 51 individuals. 51 because that's what the number, it's because people have been using that number since the beginning, so people keep using that number so that they can compare. It just allows us to compare diversity in samples of different sizes. In the abyss, diversity can be quite variable, very low to very high. In the Pacific, diversity tends to be quite high. So that's not so good because it means there's lots of species, lots of rare ones, as Gordon alluded to this morning. Vents, I couldn't find values of expected number of species, but I'm guessing that diversity is always quite low, so the number on this graph would be near the lower end of the range. So let's look into in more details at diversity in the abyssal Pacific. This is it in a nutshell. 149 species that we know of so far. Most of these have been described from the Peru Basin, so off the coast of Chile. 51 from the CCFZ. And of these 51 species, 25, I think, 25 are also found in the Peru Basin, which is interesting because that's 5,000 kilometers away. Just to look at some examples, Acanthalamus, the genus Acanthalamus. There's two species here, Acanthalamus angustus and occultus, which are found in both areas, circled in red here. But some of them are also found in the Atlantic. So they look like they are cosmopolitan species. The problem is, obviously, we don't have molecular data to check this. They just look the same. There are studies, there are suspected 45 cosmopolitan deep sea species so far. We can't be sure because of no molecular data. It could exist, some cosmopolitan do exist in the shallow water, but we also know of cryptic species. So that's to be checked in the future. And to get into more details, I'd just like to look at two studies. The first one, which Gordon already alluded to this morning, the one by Lamshed in the equatorial Pacific. What they did there is that they had 5 sites going from the equator to 23 degrees north. They found 200 morpho species out of about 3,000 individuals. And they estimate the total diversity for all these sites to be about 350 species, which is surprisingly low as far as when I read this, I thought, oh, that's almost manageable. So that's interesting. But another interesting thing they found was that the community structure is not the same everywhere. They found that in the low productivity area, the sites tended to be more similar to each other than in the high productivity area. So 2 distinct communities identified by the multivariate statistics, which means that community structure is not the same everywhere. So let's remember that. Another study, which I found quite interesting, was conducted in the French mining claim area, and they looked at 15 sites in the nodule area and the non-nodule area. But they found 325 morpho species, so more than the earlier study, and they estimate that the total diversity will be about 510 species, which is kind of similar, and again kind of low. And I just put in the sites by Lamshed here in red just for reference. So the first study was outside the CCZ, whereas this one is in the French claim area. And they also found significant difference. There's an effect, if nodules are there, the nematode community is not the same as when nodules are not there. So although we see a number of species apparently widely distributed, nematode communities are not the same everywhere in the Distal Pacific. They're affected by probably productivity gradients, but also the presence and absence of nodules. There's also other things which likely affect the community structure, things like the topography, which again, as we heard this morning, is probably more complex than we originally thought. There's also a longitudinal productivity gradient, so we will probably see gradients in the community structure going from east to west. And we'll get back to this, I'll get back to this in the last section of the talk, because that's relevant. Now for a deep sea vents, the picture is very simple. This is it right there, 16 species that we know of with the name from around the world. Most of them from the mid-Atlantic ridge and the East Pacific rise, and a couple from the Southwest Pacific, and one in purple which occurs in both the Atlantic and Pacific, apparently, based on morphology. Now I'll show you what this looks like on a map. This is a map of the world with biogeographical regions identified based on the fauna of vents, but not nematodes, other animals. And each different color it's got the different fauna. And this is the data that we have on nematodes. So we have 7 species from this East Pacific area, 8 from the Mid-Atlantic, and 2 from the Southwest Pacific. So it's not a heck of a lot that we know about. And that leaves several biogeographical regions where we have no samples or no record of species at all. There was an interesting study by Van Roosel who looked at the genus composition of nematodes and seeps and vents. And they found no evidence of a split between the Pacific and the Atlantic in terms of genus composition, which I thought was interesting. Obviously, the story will be different at species level. And like I said earlier, the diversity of nematodes at Ghent is almost always quite low. There was a study conducted by Copley et al. Where they found on the East Pacific rise, and in total they found 17 species, which is really not much at all in dense habitats. And they also found that the whole area belonged to the same biogeographical region based on morpho species. One thing I forgot to say is that the analysis by Van Roosel et al. They see signs that the habitat within the vents, may be mussels or riftia, is more important in affecting the community structure than the distance between the sites. So what does it mean in terms of for deep sea mining? Obviously I can't give you an answer today, but I just had a go at it, and then there's some concepts that we've been playing with in New Zealand which I thought might be applicable or useful for this problem. In the CCFZ, like I said, we have high diversity, patchy distribution, lots of rare species, a total diversity of about 500 species plus. That's probably conservative, but most of these species are yet to be described, and we also know some species seem to be widely distributed. So does that mean that geographical distance is not a barrier to dispersal? So if that was the case, species turnover should not be a strong function of distance. And of course, we don't have data to check this for nematodes. But I just thought I'd give you an example from New Zealand, where we've got samples that are situated on the continental slope at a distance of up to 2,000 kilometers apart from each other. And I looked at species turnover against distance. And what this says is that as you increase the distance between sites from, say, about 1 kilometer, 5 kilometers to 2,000, the increase in the average species turnover is very small. So over that distance, geography does not play much of a role in species turnover. If you do look at the same data, looking at not distance in terms of kilometers, but distance in terms of difference in sediment characteristics, then the slope gets steeper and the fit of the line gets better, which means that the environment filters the communities a lot more than distance does. And it's only sediment. I could have added a whole bunch of other variables, but I didn't have time to do it. So what this suggests, and I'm not saying it's the case for the CCFZ necessarily, but it might be that in the deep sea, the environment basically filters what kind of community you end up with at a particular place. And the community that you have in one place also determines the functional attributes of your community, and that's a very important point. So the question that I'm asking is, could we use an approach where we try and conserve communities as opposed to individual species, as a way of bridging the gap in the knowledge that we have and will always have, and the things that we're trying to do, which is mining. So this is an example from the continental slope of New Zealand. There's an area which was of interest, it still is of interest, to phosphorite nodule mining. We didn't know much about the communities there or the taxonomy of the animals that live there. So we took some samples in a sort of block design, a bit like the CCFZ, I suppose. And we took box scores, look at the macro and fauna. So this is not nematode. It's just an example of the approach. We process the box scores, you get community data, you feed that into a software, primer, or whatever, and this tells you how many communities, distinct communities you have in your samples. And you can assign each site to community A, B, C. In this particular example, we had 5 communities. And these can be described in terms of what taxa dominate and what functional types dominate as well. Once you've identified the communities, you can try and figure out what is the link between the presence or absence of that community at a particular place and environmental variables. So it could be the presence of nodules, and in this case, community D was, that's using boosted regression trees. In that case, community D is strongly related to the presence of nodules and other things. But once you've done that, it means you can try and predict the distribution of your communities in that area using a model. So you have imperfect information and try to pretend you have perfect information. And then obviously you need to check your model. But that's the idea. So this gives you information on which you can base your management decisions. And there's a few advantages to using this technique. First of all, you don't need species identifications. You can do it at, like for this particular example, we mainly use family ID level. You could use genus level as in nematodes. Rare species don't really come into it, and that's a weakness of it. But the community distribution tells you where the barriers or the boundaries between communities exist. It's really hard to tell where one community begins and another one begins in the deep sea, especially in the abyssal plain where everything looks more or less the same. But the species tell you because they don't always like the same conditions. It's easy to model communities, so you can try and predict communities, but it's very hard to model species, especially when they're rare, as they are in the abyssal plain. Lots of zeros in your data sets, and that makes it really difficult. If you conserve the community, you can be sure that you conserve the functional integrity of the whole community, which is big, really big, probably the biggest advantage of using this technique. You will be sure that you won't lose function of your community. And you can do the same with molecular data. You can cross-check whether your approach has the same results as one based on an environmental DNA, which could be an interesting exercise. So that's all I'll say about that. I'll just move on to the vents. The situation there is quite different because we're looking at worldwide situation. We can't assume that there's propagules going from one biogeographical region to the other. So each should be treated separately, at least to begin with. It's likely that species have a restricted distribution. But diversity is so low that I think personally that there is hope to describe all of the diversity of nematodes at vents, at least for some of the habitats of the vents. So I think it's worth having a go at it. And we also need to know about whether there's species that prefer one habitat within the vents and others. And another remaining question is whether we truly have species crossing boundaries between ocean basins or not. Some people think that they're the same, but it could be that if we use molecular tools, we'll find that there are different species or different populations or cryptic species and so on. So I think that's it. Thank you for listening, and I'll happily take questions if we have time.
Time for questions?
Yep. How did you discriminate those different communities on the basis of the data that you got at the family levels? The only reason I ask is because obviously at the family level it might be easy to put those communities into boxes, whereas maybe from a generic or species perspective, you might find that those groupings may become blurred a bit more.
Yeah, it's arbitrary to some degree. You can choose to have more or fewer classification or fewer communities. So the decision comes down to the nature of your data and what you can manage in terms of how many different communities you can accommodate.
Can I ask a question about the modelling? Because I think that's a really interesting perspective. How rigorous was the sampling to generate the boundary levels?
How rigorous was the sampling?
To create your communities, you have to have a fairly good spatial...
Yes. There was a fair, like on the map, there was, I think, 30-odd box core stations, and the area was about 50 by 50 kilometers, roughly.
Okay.
But it comes down to the quality of your environmental data, which needs to cover the entire map.
So did you target particular aspects of the environment? So when you did your environmental survey, so you've got your different habitats?
The main consideration was to cover the entire area as evenly as possible, but one could try and target. If you've already got some data to say that some areas are different in terms of environment, you could try and do that.
So the sample by box score or multiple score?
Box score.
My question is related to the sampling and event sites. Usually either you sample soft sediments because you have the ROV that can allow you to collect the soft sediments, but otherwise it is a washing of mussels and tube worms that you have. And it depends on how you treat it on board. If you go through a very white screen, then you lose everything from Meiofauna.
Yeah.
So maybe there is a lack of knowledge because we don't have enough samples. The number of species that you are citing is just the result of the sampling methods.
Could be, yes. I've never sampled advance
I have.
personally, but yes, and others have done more so.
Yeah, I
so.
have.
But the thing is, I think the low diversity might be a result of just the substrate not being your typical subsediment substrate.
Yeah, it's rocky substrate, so it's difficult to sample. I can tell you.
So you saw the decrease in diversity with death.
Yes.
It's rather unusual. It's not what we find normally with animals, with organisms. Is this found at the genus level or species level, or?
This is all the data I could find on species, nematode species diversity expressed as expected number of species per 51 individuals. Yeah. But it hardly goes down to about 4,000 meters. It's only beyond that it starts to really go down.
So the paper you talk about of Costa from 1993, that is his PhD, and that is the reason that it's not in the literature or what. It's enormous big PhD.
Yes.
Made up from the Peru basin
Yeah.
of the northeast there. So that is one that not count, really?
Well, not in the eyes of the code of zoological nomenclature, but in practice, everybody uses it.
Of course. And do you know how he get all this nematode out? He crossed the kind of sound.
No, it's soft sediment. And
So-
it's not. It's not from the nodules. It's from the. It's
But
called,
he's also one from the nodules.
yes, but they were probably washings.
Okay, only washing. So he's not taken out because the Meiofauna is inside.
Some of it is.
Definitely inside.
Some of them are.
Yeah, all the database, of course, is inside. I mean,
You have to.
yeah, you have to really to squeeze them.
Yeah.
I think
Sure.
in the early days people didn't do that.
Yeah. Yeah.
Well, you have to-
I think this is one of the things you're going to have to explain to us
Yeah.
in your talk.
How come up that late?
Huh?
But I think that is less the case for the nematodes. I mean, you find some nematodes in the nodules, but there are very low numbers. So it may be different. It will be interesting to find out about the target rates and the
Yes, yeah.
calories.
Yeah.
But for the nematodes, they're not really into the
It's
nodules.
not inside. Okay. Yeah.
Sorry, it's just a minor question. You might have told us. I really liked your modelling of the communities and your predicting business. Which one of your 2015 papers was it in?
That paper came in DSR this year. Sorry, did I hear right?
Yeah, I
No, that's like DSR.
Deep sea research.
Right, super.
Of course. Of course.
Yeah.
He's Googling it as we speak.
Yes.
My comment related to Anne's reply is when you're sampling in vents and you collect soft sediments and that they have bacterial mat, our colleagues from IFAMER can confirm that when you core, usually the bacterial mat will sink. It will sink down to maybe 10 centimeters. So sometimes some nematodes will be there because they are associated to the bacterial mat. You have done that too.
I collect
Yeah.
the bacteria mass.
And then inside the core you will see an increase with depth instead of having all the abundance in the surface.
Yes, I'm wondering about the bioindicators function of nematodes. For example, in the freshwater or shallow water, you often use nematodes as an indicator of organic enrichment. Do you think something similar would work in deep sea? Or?
Yes, I mean there are genera which are typically associated with organically rich habitats in shallow water. So we can take a guess in deep sea that these taxa are also associated with organically rich areas.
You don't have any evidence.
Well, yeah, there's like Halomonistra, I think, in seeps are very highly abundant.
But I think the evidence is little, but the study by Marina and Dimitri Milutina, who sampled in the 36-year-old track of an experimental mining exercise, and they compared with the fauna outside the track, and they identified differences in communities, although both were sampled in sediments, but one was a disturbed sediment 36 years ago, the other was an undisturbed sediment. And the communities differed in the sense that you had some shifts in dominance. It was not that you had completely different species. Some species became more abundant than others.
Oncolemus was highly abundant in the disturbed track.
And there was more organic matter in the track, if I remember well, than outside. And Oncolemus is indeed one of these bigger nematodes that is considered as a kind of omnivore, scavenger, predating behavior. So it makes sense to assume that indeed these scavengers, predators, which are more mobile, may behave differently in disturbed areas. But the evidence is little. It's like 4 samples. And these are things we did repeat during the Zona cruise, and where we hope to get more evidence.
Other questions? In which case,